By Helen Briggs. BBC News Media playback is unsupported on your device. Media caption footage courtesy of Qiang Sun and Ma Mingpu. Chinese Academy of Sciences. 2 monkeys have been cloned using the technique that produced Dolly the sheep. Identical long-tailed macaques Zhongzhong and Hua were born several weeks ago at a laboratory in China. Scientists say populations of monkeys that are genetically identical will be useful for research into human diseases. But critics say the work raises ethical concerns by bringing the world closer to human cloning. Qiang Sun of the Chinese Academy of Sciences Institute of Neuroscience said the cloned monkeys will be useful as a model for studying diseases with a genetic basis, including some cancers, metabolic and immune disorders. There are a lot of questions about primate biology that can be studied by having this additional model, he said. Image copyright Chinese Academy of Sciences. Image caption Zhongzhong was created by somatic cell nuclear transfer. Zhongzhong was born 8 weeks ago and is named after the word for Chinese nation or people. Hua was born 6 weeks ago. The researchers say the monkeys are being bottle-fed and are currently growing normally. They expect more macaque clones to be born over the coming months. Not a stepping stone. Prof. Robin Lovell Badge of the Francis Crick Institute, London, said the technique used to clone Zhongzhong and Hua remains a very inefficient and hazardous procedure. The work in this paper is not a stepping stone to establishing methods for obtaining live-born human clones, he said. Watch Dolly. The world's most famous sheep. Prof. Darren Griffin of the University of Kent said the approach may be useful in understanding human diseases, but raised ethical concerns. Careful consideration now needs to be given to the ethical framework under which such experiments can, and should, operate, he said. Dolly made history 20 years ago after being cloned at the Roslin Institute in Edinburgh. It was the first time scientists had been able to clone a mammal from an adult cell, taken from the udder. Image copyright Science Photo Library. Image caption Dolly the sheep was the first mammal to be cloned 20 years ago. Since then many other mammals have been cloned using the same somatic cell nuclear transfer technique SCNT, including cattle, pigs, dogs, cats, mice and rats. This involves transferring DNA from the nucleus of a cell to a donated egg cell that is then prompted to develop into an embryo. Zhongzhong and Hua are the first non-human primates cloned through this technique. In 1999, a rhesus monkey embryo was split in two in order to create 2 identical twins. One of the baby monkeys born through that technique, called Tetra, has the title of the world's first cloned monkey, but it did not involve the complex process of DNA transfer. Much failure in the study, published in the journal Cell, scientists used DNA from fetal connective tissue cells. After the DNA was transferred to donated eggs, genetic reprogramming was used to alter genes that would otherwise have stopped the embryo developing. Image copyright Chinese Academy of Sciences. Image caption Hua was born 6 weeks ago in a Shanghai lab. Zhongzhong and Hua were the result of 79 attempts. 2 other monkeys were initially cloned from a different type of adult cell, but failed to survive. Dr. Sun said, "We tried several different methods, but only one worked. There was much failure before we found a way to successfully clone a monkey." The scientists say they followed strict international guidelines for animal research, set by the U.S. National Institutes of Health. Co-researcher Dr. Ma Mingpu, also of the Chinese Academy of Sciences in Shanghai, said, "We are very aware that future research using non-human primates anywhere in the world depends on scientists following very strict ethical standards.